4 sheets. If you came in late, you might not have your sheets. I don't know. You should have 4 sheets. Now, we got a lot to do today. I think you have, what, 21 pages of notes for today's lecture? The good news is you will not be, you won't make it through all that. We'll be finishing up some of it next week. So, what I want to do is help you, hope, to organize this. So to take this large number of pages and really get it down to about 4 pages. That's what these 4 pages, this diagram is for. So, we did talk previously, didn't we, about the difference between glomerular filtration, tubular reabsorption, tubular secretion, we've done all that. And what's filtrate, what's tubular fluid, what's urine. We talked about that as well. Everybody's good? Okay. Now, on your notes, page 3, you should have a figure that looks like this one. Okay? What we're going to do is we're going to take notes, and I'm going to get rid of this in just a minute. We're going to look at our drawing, just our drawing. All right. So, let's turn this off for now. So you have your notes. We'll go back and forth between a little bit, but mostly we're going to be on these pages. So this is a challenging lecture. If you've already done your notes, you know it's challenging. So what I'm going to try to do is to help you to make it make sense. Now, when I presented it in the lecture, I took each substance, sodium, went through the whole way, through the tubules. Then I took the next substance and went, we're going to do it a different way this time. Hopefully, it's a different way of approaching it, and it might make it easier for you. We'll see. So we're going to take each section with each page there that you have and look at what's happening in these tubules along the pathway by section. So we have the afferent arterial, not on your figure there. So you should have which figure now in front of you?
Obligatory water reabsorption.
Right. Obligatory water reabsorption. So this part in glomerular filtration is what we looked at the last time. So you have your figure already. So my students in the past told me that they preferred to have a drawing, something already there, so they didn't have to actually draw it, because it's plenty even without having to draw it. So that's why you guys have a drawing.
Thank you.
Oh, you're welcome.
You're the greatest.
I love that. You can keep it up. We're going to note a few things relative to the relationship of all these, the anatomy here, and what's going on. Right? So we know that this blood vessel up here that actually wraps around the tubule, what is this blood vessel?
Peritubular.
Right, it's the peritubular capillaries. Now, when filtration took place, we lost fluid, right, water, but we retained the proteins, right? So this fluid that comes out, this blood that comes out the afferent arterial, has 2 properties that are significant. Because it's lost the water, it has a relatively low hydrostatic pressure. And remember, that's the pressure that's forcing things out of the capillaries. So it's not much push out. In addition, because it retained the proteins, it's got a relatively high cup pressure. And that's the colloid osmotic pressure. And that's the pressure that's helping to pull the fluids into the capillary. So we have very little pressure pushing out, and we got a pretty high pressure pulling in. And so there's this sort of setup, right, if you will, to help to pull the fluids and material back into the peritubular capillaries. That's one idea that we want to look at. Another idea that we want to look at is the fact that we have, when this fluid is first here, it's filtrate, right? What's it called once it enters the tubule? It's tubular fluid here. Now, there's 2 ways, if you will, that substances can move across between these cells or over across the cells. So this one, if it goes between the cells, which we're really not gonna talk too much about, but because it goes beside the cell, this movement is referred to as paracellular. Para means beside. Remember, like paranephric fat that we talked about before. So paracellular transport. As opposed to if we want to move a substance, we're talking mostly reabsorption today, move it across these two membranes, then that's actually crossing it, so it's called transcellular. Everybody all right with that?
Oh, yeah.
So we got paracellular, we got transcellular. Because most of what we're going to be looking at is transcellular, when we look at this cell, we actually have two membranes that we've got to account for. This one that's next to the lumen, right, this space here. And so this is called the luminal membrane. And this one here is what?
Basal.
Basal. So we're going to see that to move these, for these substances to move, the process involves membrane transport, stuff we talked about since last semester, right? So simple diffusion, facilitated diffusion, active transport, chemosynthesis, all those can be involved in this movement. All right, let's begin looking at our very first idea, which is what's some of the substances that we have to reclaim, and one of those we're going to look at first is glucose. So you have to add that to your figure. This glucose, we got glucose, and we're going to look at how glucose has to cross. It's going to use transcellular mechanism, right, in terms of the method to get it across. So we have two membranes that we've got to look at. So we have to know where is glucose relatively high. Glucose is relatively high inside of our cells, of these tubular cells. That's important because you got to know if you're moving the substance up its gradient or down its gradient when it crosses the membrane. So that means if you know that it's high here, obviously then it has to be low on either side, right? Outside the cell will be relatively low. So I say to you, okay, we got to get glucose from in the lumen, and we want to reclaim it, right? We just filtered it, and it's nutrients, it's good fuel. We don't want to urinate it out for lots of reasons. So we have to have the body is going to be able to bring it across that first membrane. Is it moving up the gradient or down the gradient? Is it moving from low to high or high to low?
Because we said it's relatively high.
So it's relatively low here, where it's coming from, relatively high inside the cell. So it's moving up its concentration gradient. So is it going to engage in an active process or a passive process? Active. So we know it's active. So when we remember our work from before on active processes, really you have two: primary active transport, when you want to move small molecules, primary active transport, and secondary active transport, which simply means you use ATP or you use something else, right? And so as it turns out, the process is not primary, it's secondary, right? It's secondary active transport, AT, active transport. So in secondary active transport, remember there's the movement of a second substance to get the glucose into the cell. And as it turns out, the substance that's going to be moving down its gradient this way is sodium. Right? So here we see this idea that we've seen several times now. And this protein is the sodium glucose symporter. And the process it's engaging in is symport, right? Secondary active transport, specifically symport. So we're allowing the sodium to come in the cell. As you'll see, we don't yet know why, all right? We'll just put it in there for now. But if we remember our work from neurons and other cells, sodium is always high outside of cells, right? So sodium is moving down its gradient to move glucose up its gradient by active transport. Everybody good with that first one? No problem? Because if you get the rhythm, it's not too bad. Now, once this glucose is moved into the cell, glucose has to be moved across this other membrane across the basolateral membrane. It always has to cross two membranes. So there's usually different processes to move across the two membranes. Now we're allowing the glucose to move from where it's high to where it's low. Passive or active? Passive. So what do we have in passive? We've got really basically two types: simple diffusion, facilitated diffusion. The only thing that can move by simple diffusion is small and nonpolar molecules, right? And glucose is not that. Glucose is polar. Remember, it dissolves in water, sugar water, right? For example. And so this process here will be facilitated diffusion. So it just has a carrier protein. Remember about carriers. It has a carrier protein that's going to carry the glucose across. Instead of a channel, because remember channels are for ions. So this would be specifically a carrier protein that's going to help this glucose move across that second membrane. So far, okay?
Yeah.
So what percentage of glucose do we normally reclaim? We reabsorb?
100.
100%. And that is just a representative of any of the nutrients. Could be amino acids or whatever. We just use glucose as our example. Now we're going to go back to the notes just for a minute or two and go over to page 4 of your notes. Because we want to look at a concept, a related concept. I think it makes more sense after we talk about glucose movement and its reabsorption, and then talk about these next two ideas.
So on this diagram where it says A and B, that's where you wanted us to write secondary active transport
there's a blank spot.
and facilitated.
Yeah, I should have mentioned that to you. That's a good question. So on, I wish I had glasses on. I could actually read my writing.
I have all the.
A is secondary active transport. B is facilitated diffusion.
Do you
And
want
then the
me-
blank up there with the percent is 100%.
Right.
No, I've got some.
Sure, okay.
Thank you.
So A is secondary active transport,
Uh-huh.
B is the facilitated.
Diffusion, and then the blank up there with the percent is 100. This figure should be so familiar to you now, right? So what we're looking at is this idea of what's called transport maximum. Transport maximum. Now, transport maximum, if we're looking at relative to reabsorption,
This isn't on one of our pages, is
this is
it?
on page 4.
Oh, okay, so it's
Page
not
four,
one of
yes.
these, okay.
So we went back to your notes, page 4. Transport maximum is a rate. How fast can the nephrons do something, right? And so the units of measurement are typically given in milligrams per minute. And so transport maximum is asking the question, at what rate, what is the maximum rate that the kidneys can reabsorb a particular substance, right? Or secrete, but in this case, we're really much more concerned about reabsorption. And so, for example, if we look at transport maximum for glucose, sometimes the value that's given is 320 milligrams per minute. So that's the maximum rate. 320 milligrams every minute can be reabsorbed by the kidneys back into the bloodstream. So, sort of reminds me again of that show, the I Love Lucy episode, right, where you've got the material coming down, it's been filtered, and we filter, let's say our glucose will make it green, gets filtered into the tubule, and by the time we're done here, all that glucose should be been reabsorbed back into the bloodstream with none of that left, zero left to be excreted in the urine. That's normal, right? As long as we don't exceed the transport maximum.
I have it as, from our notes, 375.
Yeah,
Does it
depends on-
matter? It's just.
No, I would never ask you the number. Depends on what resource you look at, what number you'll get.
Okay.
Now, everybody good with transport maximum? A related idea is renal threshold. Now, the renal threshold is not a rate. The renal threshold is actually a concentration, which is, in this case, milligrams per deciliter. So each substance, so the value they give again is, I don't know, about 300. You don't have to worry about the specific number. So whatever the concentration is, the normal concentration, that plasma concentration for glucose is the total concentration we can have of that substance before it will exceed the transport maximum. So we have a certain amount of glucose concentrated in our blood, right? Let's say this is under 300. It gets filtered at the same rate, right? And these little protein guys over here, right? These little carriers and symporters can reabsorb that and no problem. And it takes all of it out and we're left with zero. But what happens if you have eaten a really big candy bar or you're diabetic? Concentration of glucose, either temporarily or more permanently, is greater. So glucose levels are rising. So what happens then, of course, is we get more filtered and exceeds the transport maximum. And what happens, the critical thing then, is glucose is not 100% fluid. Now, obviously this is bad, right? You don't want to lose your fuel by your urinary system. But there's a double whammy. Glucose is a solute. It also pulls water with it, and we lose more water. So one of the symptoms most people will figure out that they're diabetic is excess urination. And then, because you're urinating more than you should, you're losing more fluid, you're also thirsty. And that's because the glucose is acting with an osmotic force to pull the water with it as it goes out with part of the urine. Everybody good? Okay, so we're back to this figure over here. Just wanted to show you how that relates to this. Now, we also have some other substances. Now remember, small amounts, scant was the word they used. Small amounts of proteins can get filtered, right? Now, if I say to you, are proteins normally filtered, what would you tell me? No, they're not, right? But there is a small amount that's going to come through. And so these proteins, remember what a protein is. A protein is a polymer of amino acids. It's not a tiny little thing like glucose. Glucose is one little monosaccharide. We're talking about, relatively perhaps long strands of amino acids. So there's no channel, no carrier that's going to be able to take that protein and put it in on the other side. So instead, the protein will be up here at the surface. And how will that cell be able to pull that and bring that protein in? By what process? Pinosynthesis.
So what blank are we writing?
So the blank, for those,
I see.
yeah. So you could put pinocytosis or just more broadly endocytosis. Now this protein now ends up inside of a vesicle within the cell. It will fuse with that organelle you know as the lysosome, and it'll chop it up, right? Break it down. Always helps to have all the diagrams. And so these are now amino acids, which are relatively small molecules. And how will they be dumped then on the other side? By what process? Exocytosis. So that would be, in terms of your diagram there, you would have D would be amino acids, that's what's actually being released, or whatever, and exocytosis would be the process, or vice versa. Okay. Now sometimes, there's another way this might happen, is there are actually enzymes embedded in the plasma membrane, the luminal membrane, and they can chop up the protein before it even enters, and the amino acid gets in. It doesn't matter. You're going to get proteins from this side, break them down, and release amino acids on the other side. And then those amino acids get absorbed. What percentage? 100%. So that would be the percentage there by protein, 100%. All right. Doing okay? So we move now to our next substance, which is going to be sodium. Now, sodium, remember, very little here. Sodium, Na+, is an ion, tiny little guy. high, right? And we want to, the body wants to be able to reabsorb it. So where is sodium always high? Outside the cell or inside the cell? It's always outside. So that means that in terms of these cells, we would see that there's relatively low sodium, which we already know, inside cells. And that's true, as far as I know, all cells in the body. So the question becomes now is, by what process, so it's going to be relatively high here, right? By what process will sodium be moving across that first membrane, the luminal membrane, active or passive? Passive. So it's moving down its gradient through some process, right? It might be as part of the Symport movement. It could be a little channel that it moves through. There's lots of varieties. We're not going to, whoa, no way we're getting all those varieties. But we know it's a relatively passive process here. Because the substance, in this case sodium, is moving down its concentration gradient. So there's a variety of proteins that will allow that movement to happen. We just saw one of those right here. All right? So sodium's in. So now I'm going to move from where it's relatively low to where it's relatively high. Active or passive? We know it's going to be an active process. How do we pump sodium? Always. Pump? What kind of pump? Sodium potassium. So we're going to pump how many sodium? These pumps do. How many? Three sodiums out of the cell for every what? 2 potassiums that come in. All right, so we call this, as we know, this is sodium potassium pump. Now, what percentage of sodium do we reabsorb? Not 100. About 65% of sodium is reabsorbed. So that's about, I like to think in fractions, so 2/3. So about 2/3 of your load has been reabsorbed. Not all of it. That's critical because one of the jobs of the kidney, which we talked about the very first day, is to regulate the amount of sodium and other electrolytes that are in your blood by just changing the amount that we lose in the urine. So, but you say to yourself, well, you know, you just dumped a whole load of sodium. You're just going to reclaim right here at the beginning 2/3 of it. Now remember, you can see from all this, there's a lot of reabsorption going on. What characteristic does that luminal membrane have to assist with this? Anybody remember? When you want to increase reabsorption or surface area. It's got microvilli. I didn't draw them on there, but remember they're there. So they have a lot of microvilli here.
When you say the 65% reabsorption, that's just for the PCT, right? Because
correct. That's
okay.
just PCT. Okay. So we're working by section. So we're in the proximal convoluted tubule, which I probably should have written up here. Right? Just to remind you. So we're in the very first section, proximal convoluted tubule. All right. That looks good. So on F, you should have facilitate diffusion or something, like passive process. Could be lots of different things there. G is active transport with the sodium potassium pump. All right. Now, we have water. So, water always follows what? So wherever sodium goes, water follows along. By what process? So it's across both membranes by osmosis. So the sodium goes, water. So it's osmosis. Now, what's it need to actually go across? We're going to see there's proteins. We talked about these last semester. That are pores for water called aquaporins. Remember those? I don't know.
Yes.
Aqua water, pores, holes. So we got aqua. The process there on H and I would both be osmosis. How much of the water do we reabsorb? If you had to make a guess, you guess, well, if we reabsorb 65% of the sodium, we're going to reabsorb 65% of the water. Okay, so H2O here. Now, what they'll say is the water was obliged to follow the sodium. So they refer to this then as obligatory water reabsorption. We really don't have control of it in terms of physiology. Sodium goes, the water goes behind it. All right? Now, just so you know, and I'm not going to ask you about this on your test, but all the other electrolytes, like most of the electrolytes, like chloride, magnesium, and phosphate, and sulfate, and all these other ones, it's interesting what happens. When the sodium's a leader, sodium goes, water goes. But what happens to the relative concentration of the remaining solutes here, if we lose water, what happens to the concentration? Relative concentration goes up. So we have a concentration gradient for all those electrolytes, so they're going to move by a passive process across that luminal membrane. Now the interesting part is, when water moves from here to here, it also makes it more concentrated here than here, but moves across the second membrane also by a passive process. So it ends up sodium goes, then water goes, then the other electrolytes go. Last thing here relative to the proximal convoluted tubule is we have a hormone, which we talked about last semester, called the parathyroid hormone. Parathyroid hormone actually has two targets in the kidney. One of those is the proximal convoluted tubule. And what it's going to do is it wants to block, it actually inhibits the movement of phosphate ions. So if parathyroid hormone binds to these cells in the PCT, it's going to inhibit the reabsorption of phosphate. Now, you're like, what? Why would it do that? I thought, if you're thinking, well, I thought parathyroid hormone controlled calcium, right? We release it when we have low blood calcium. It's supposed to help us to retain calcium. Here's what you got to understand. Phosphate and calcium want to go to bed together, right? And so, you got all that? Sarah just looked up. So we got phosphate, we got calcium. If phosphate and calcium find each other in the blood, they're going to go to bed in the bone. So if I want to keep calcium levels up in the blood, which is what the problem was low blood calcium, I got to get rid of its partner. That's what we're doing. So we're inhibiting, by way of parathyroid, the ability of phosphate to be reabsorbed. We're going to lose that phosphate in our urine, and that lets more calcium be free in the blood and continue to circulate. So that's why it's got to inhibit phosphate. Okay. Now, we got our first diagram done. You all right? You'll be glad to know the second one's not as big, if your head's starting to hurt a little bit. So we're going to the second one, and that's the one of the nephron loop. Like this. Now, on the nephron loop, really, there's really one critical thing I want you to understand about the nephron loop. This more minor thing is that all this that was going on here relative to ions and water and all these electrolytes is going to continue in the nephron loop. And those materials are now pulled into the vasa recta instead of the paratubular capillaries. So additional amounts of these substances are being reabsorbed. But why do we have these long nephron loops is that we can establish a concentration gradient out here around the tubules. Probably should have done a different color. is, outside the tubule now, is a concentration gradient of mainly, if we had to figure out one substance, we'll say sodium chloride, right? So you gotta imagine now we have a salt gradient. And so the further we go down to the tip of the nephron loop, the more concentrated it becomes. There's a whole section in your book that describes how that concentration gradient is established and maintained. You're welcome to read it, all right? I'm not getting into that. We have enough issues trying to understand some other things relative to this topic, all right? But what I want you to understand is this. That gradient is necessary for the hormone ADH to function. You ever heard of loop diuretics to control blood pressure? You will, all right? Loop, what are they talking about, the loop? That's the loop. That's the nephron loop. And those diuretics, they interfere with establishing this concentration gradient, and so ADH can't function. And so what do these people do that are on loop diuretics? What's their urine output like? You want to get rid of fluid if you have high blood pressure, or keep it? Keep what fluid? Yes. You want to? Yeah.
Yeah, you wanna get rid of it.
Yeah, you want to get rid of it, right? Because if you get rid of fluid, what happens to your blood volume? It goes down, blood pressure goes down.
Food, and I'm like,
No, fluid is fluid.
Sorry, I'm coloring.
So, that's all right. Okay. Everybody good? We'll come back to that idea later. That's all I want you to know on the second page. So we're done the second page.
it says why ADH to function
Yeah.
in CT and CD, CT means.
Yes, yes. So
Convoluted both in
we're going to
proximal and
see.
distal?
So when we go into the last section, this next page, the third page, we're going to look at that ADH and how that gradient helps us. Okay? So for right now, all we need to know that there's a gradient there that's established by the nephron. All right. Any questions before we go to the third page?
This one looks like a board game.
Yeah. All right, non-acid base, that's our fourth page. We want to go to the third one that was sitting like this. Most confusing page. Hopefully by the time you're done, it'll make sense. We'll see. All right. Gotta do our drawing. You can, I've actually tried to line these up so you could actually take them together if you so choose to do so. If you'd like to have it all at a glance. So this is the distal convoluted tubule, DCT, which we know goes into collecting tubules and collecting ducts. Urine eventually is going to go out here. Okay? See where the lumen is there? Coming down like this. You with me on the diagram? You all right? Now, when you think of the distal convoluted tubule and the collecting tubule and the collecting duct, what I want you to think of is totally different really from what's going on with the proximal convoluted tubule. The proximal convoluted tubule is saying, oh my gosh, we just lost all this good stuff. Let's try to get back as much as we can, right? That's why the microvilli here. This area of the tubule are really where we can control the relative amount of a substance that's lost. Now, would that apply to nutrients? No, we've already reclaimed all and reabsorbed sodium, glucose and all the amino acids and proteins. What we want to do in this section is really regulate electrolytes in water and how much is actually lost. So in your notes there, we're going to first, though, talk about on letter K there, see K? That is going, we're going to continue that discussion of calcium. And so parathyroid hormone also talks to the cells over here in the DCT and is going to stimulate the reabsorption of calcium back into the capillaries. So this is stimulating this. So we reabsorb calcium. So we're always gonna lose calcium as part of our urine, but we wanna decrease the amount that's actually lost.
So it stimulates calcium to reabsorb.
In the DCT, in the distal convoluted. So it's blocking phosphate reabsorption in the PCT and stimulating calcium reabsorption in the DCT, in the distal convoluted. So where's calcium end up? Calcium is hopefully reabsorbed back into the bloodstream. Now, these cells over here, a couple cells that we're going to look at. One of the cells we're going to look at are the principal cells. Sorry, might not want you. Just try.
So for K, we put parathyroid hormone
Okay.
or calcium
I
reabsorption.
was thinking potassium.
I'm
K,
sorry.
yeah, PTH. Yeah,
PTH.
parathyroid hormone. For K, letter K. Now, we have three major hormones that are of interest here. We're gonna, the one we've already done, that's the parathyroid hormone. We also have aldosterone. Right, now, gotta just remind you of a few things or ask you a few questions as we get going here. First question is, what is the source? Where is the endocrine gland that releases aldosterone? Adrenal cortex, right? Is it, what kind of hormone is it? Is it water soluble or lipid soluble? Yeah, it's a steroid. Remember the ones that are reproductive hormones plus the ones from the adrenal cortex. So this is a steroid hormone. That is significant, right? Because we have to remember what we know about hormones and steroid hormones. Where's the receptor for steroid hormones? In the plasma membrane or inside the cell? Steroids are small and nonpolar molecules, so they come right in, and so the receptor would be inside these cells. So we'll make aldosterone, make it red. Okay, so say it looks like this. So it comes on in and binds to the receptor, and you get a hormone receptor complex. Now, let's see how good your memory is. What do steroid hormones do once that complex is formed? Make new proteins. So how are they going to do that? Somewhere in here is this nucleus, finds the DNA, and makes that cell make new proteins. So it's synthesizing new proteins. So the question becomes is, just so you get your bearings, here's the luminal membrane. Here's the basolateral membrane. Some kind of protein channels are here, right, for sodium, right? Whatever it might be. We'll just put sodium channels. So it's making more sodium channels. That's the protein, because channels are protein. What particular substructure is being made and put on the basolateral membrane? Based on what we already know from over
Sodium,
there,
potassium pumps.
sodium, potassium pumps. So we see sodium, potassium pumps. And we get more and more of those. So that's what it's doing. So for every additional pump that's placed out there, each one of those pumps is doing what with sodium? Pumping it out or in? Right here. Where's sodium low? Inside, high outside. Which way we pumping sodium? Always
Out.
out. How many?
3.
What gets pumped in?
Potassium.
So you're saying the aldosterone causes the formation of the pumps?
Yes.
Okay.
And these channels.
And, okay.
Go. Because we need them on the channels or whatever protein can help us over on this
So
side.
are we writing these under the net effect then?
Yes, I'll get that in just a minute.
Okay.
So the sodium is moving through these channels down the gradient, let's say it's a channel, and then they're going to be pumped up the gradient. So what if I said to you, what is the net movement of sodium? Did we reabsorb it or secrete it? So we reabsorb, would you agree, sodium? Sodium is reabsorbed. Everybody all right with that so far? What always follows sodium?
Water.
Water. So water comes along for the ride. So what else do we reabsorb? We reabsorb water. So water is also reabsorbed. But at the same time, what's being secreted, because over here we see these as well.
Okay.
Yeah, K is going this way. We're losing potassium. That's critical to understand. So there's this loss. So potassium is being secreted in order to be ultimately excreted
To the chop block.
to the chop block. It's going out, right? Going down the tutor. Or pooter, or whatever. Now, on your notes there, net effect of aldosterone is three things, NO and P. Sodium reabsorption, water reabsorption, potassium secretion. That's critical to know that. You're going to be writing about aldosterone as part of your essays. Not this part, it's later. I'll show you how it's all integrated next week. Something to look forward to for next week's lecture. You'll all be excited. Now, what if I said to you, if you understand that, you have a patient with an adrenal tumor, and they have producing excess amounts of aldosterone. Tell me about their blood sodium levels.
Really
It's gonna be really high.
Really high. What about their potassium levels?
Really low.
Can you die?
Yes.
One of the most lethal imbalances, remember, is potassium.
So they'd also be having issues with edema, probably, I mean,
Yeah, they're gonna have all kinds of fluid problems, holding fluid. Yeah, mess up their heart rate, yeah. And we'll look at that, we do electrolyte imbalance.
So for L and M,
L and M. So L is facilitated diffusion. We're assuming it's a sodium channel. And M would be active transport.
thank you.
Now, I did have a little note there, I didn't even have you add it to your figure, it's already on the figure, is that ANP, atrial natriuretic peptide, which is a hormone we'll talk about later when we do your essays, is inhibiting the release of aldosterone. So if it inhibits the release of aldosterone, what are we doing with sodium? We're inhibiting its reabsorption, so what happens to the amount of sodium we then lose is decreased. All right, so if you want to make a note, AMP ends up causing us to lose more sodium, right, in our urine. All right, next one. Everybody good with aldosterone? It's critical to see the difference between the hormone aldosterone and the hormone ADH. They have completely different functions relative to how they operate. They too have, will bind with principal cells, but antidiuretic hormone comes from where? Where is it released from? Posterior pituitary. Remember, there's only two from the posterior pituitary, antidiuretic and oxytocin. It's coming from the posterior pituitary. What kind of molecule is it? Well, we know it's not a steroid, not a reproductive hormone. It's not coming from the adrenal cortex. It's a water-soluble hormone. So if it's a water-soluble hormone, where are its receptors? Are on the surface, right, in the plasma membrane. So ADH, we'll say this one's make, comes up here and it binds to the receptors. Now, it's not a steroid hormone. So here's what's, to me at least, is interesting. Already in the principal cells are vesicles. And in those vesicles are aquaporins in the membrane itself. So they're already made, they're already hanging out like this. Now they're not inside the vesicle, they're actually in the plasma membrane. So when ADH, remember first messengers and second messengers, when ADH binds here to its receptor on the principal cell, it's going to induce, remember that whole thing about maybe G protein and getting cyclic AMP, some second messenger system is going to be used, and it's delivering a message that says to these principal cells, hey, we need more aquaporins on the surface. So the vesicles are already there with the aquaporins. So these aquaporins will be put into the membranes. Simply by allowing those vesicles to fuse, their membrane, to fuse with the plasma membrane. So now these aquaporins, as these merge over here, actually increase the number of aquaporins. So if I say to you, what does ADH do? And I will ask you this, what does ADH do to the principal cells? It increases the number of aquaporins. That's it. That's all it does. Creates more holes for water. Did it make water move? Nope. Did it move sodium? Nope. What did it do? It increased the number of aquaporins. So, water always is pulled from an area to another area by what process? Osmosis. So the question is, what's pulling this fluid from the lumen, from the tubular fluid, into the bloodstream? What's pulling it this way? Concentration gradient. Because you got to remember, this is all not flat out like this. This thing is flipped back over, and these two are next to each other. So this concentration gradient that we would see here that pulls that water. So what happens if I get rid of the concentration gradient? What happens to my ability to use ADH to pull my water back out? It's impaired. If you drink excess amounts of fluid, one of the things that happens is you dilute that concentration gradient. Interesting, right? And so your ability for ADH to function is not as good. Interesting, I think. All right. That's it on that page. We got our last page. So your head hurting? So let's go to the acid-base balance. Now
So
what I did with
letter
this one,
N equals,
N, sorry, I was trying to fill in my leg.
it's okay,
N,
and M and
M is osmosis. N is number of aquaporins. Everybody good?
yes, thank you.
And
Now.
they increase, right? Yeah,
Huh?
they increase.
They increase the number of aquaporins. So the last thing that I saved for you is acid-base balance. And we're going to be doing a whole lecture on that and a lab on acid-base balance. You already have your figure there, right? All right. Acid-base balance is a function of two substances.
I'm not sure what you just said.
An acid, which is acid. That's hydrogen ion. And then bicarbonate, HCO3 minus, which is a base, a weak base, bicarbonate ion. That's the two things you gotta pay attention to. Now, when we filter, hydrogen ion, as it turns out, is not filtered. We're not going to get into why, it's just as, maybe I can tell you. It's attached to proteins, which are negatively charged. So the proteins are sort of protecting them from getting filtered because they're attached to this negatively charged protein. Bicarbonate, in comparison, can be filtered. Now, you got a problem. You kept the acid, right, when we filtered, and we dumped the base. So the first thing we have to understand is, how do we get the base back? We still have the acid, we're not gonna worry about it yet, but we've got this base. So the first thing we gotta do is we wanna reclaim the base. So in the tubule fluid, we're not gonna see yet but where it comes from. It's gonna bind with hydrogen ion. And when it binds with hydrogen ion, it forms carbonic acid, something we're used to. Carbonic acid, remember, is unstable and becomes CO2 and water. The CO2 is a small nonpolar molecule, respiratory gas, and it goes into the tubule. It diffuses. It's no barrier, right? Simple diffusion. It comes in. Now, CO2 then, inside the cells, these cells, hooks back up with water and forms, not surprisingly, carbonic acid. Carbonic acid is going to, remember, it's a reversible reaction, goes that way. So what did we do? What was this?
There's a big cycle.
Yeah, what's the word you would say? What happened? This equation then reverses, right? What does the bicarbonate do? It then gets put in and reclaimed in the blood. So if you're thinking, you might be like, what the heck? Why didn't bicarbonate just go across this membrane? I don't know, it just can't, right? So it has to be converted to CO2. The CO2 is small and nonpolar, it diffuses, we make it back into bicarbonate, and then we can get it across into the bloodstream, all right? We're going to reabsorb, in your notes there, is about 80 to 90% of the bicarbonate is reabsorbed at this point. So you've just lost it, and you get almost all of it back by that process. Then you go down into the nephron loop, and you reclaim the rest of it, the 10 to 20% that's still there. So when it goes into the nephron loop like this, reclaim the other 10 to 20%. So then I say to you, what percentage of bicarbonate is in the tubular fluid when it enters the DCT? 0. How much hydrogen ions in there that was filtered? None. Right? So we're back to where we started from. So where are the molecules of interest? They're in the bloodstream, right? Both hydrogen ion and bicarbonate. That brings us over now to where it actually gets regulated down here in the DCT. So we have two cells of interest, type A intercalated cells.
Quickly, before we get to that, number 1 was 80 to 90%. Number 2,
Yeah, I'm sorry.
movement.
Number 1 is 80 to 90%. Number 2 is net movement of bicarbonate then is from the tubular fluid into the blood. Should have told you that explicitly. 3, same action as in the PCD, but the remaining 10 to 20% of bicarbonate is reabsorbed. So in number 4, what's left of the bicarbonate is 0. Missy?
Yep, so to go across the luminal membrane, it has to break down and then be recreated, but then across the basolateral membrane.
It doesn't have, it gets, there's actually proteins that will assist its movement, yeah. We just don't have those proteins on that other cell. It may be that it wouldn't be able to control because the CO2 wants to diffuse anyway, so maybe that's the reason, I don't know. So type A cells, remember what type A does, what does type A want to secrete? Acid. So, here's what happens relative to this process. We're going to let the CO2 that's in the tubular fluid diffuse in again to these cells. Hook up with water, become what? Carbonic acid. Split into hydrogen ions and bicarbonate. The paired tubular fluid is over here. So what happens to the hydrogen? And we're gonna, what did type A cells do? They have the proteins that move it this way. So we're secreting acid in order to excrete acid. That's the cell that's typically active in most people. Based on our metabolism and our diet, we tend to have to get rid of acid. Right? We make more and we eat more acidic foods. The bicarbonate, which is the base, gets reabsorbed. Right? Now, I asked you a couple questions. What happens then, type A does what to urine pH? Make it more acidic or more basic? More acidic, which is doing what to the pH? Lower. What's it doing though to the blood? It's keeping it, so it's getting rid of acid, it's got an acidic problem, gets rid of the acid, and also makes new base and keeps that base. So it raises blood pH, so it lowers urine pH, get rid of acid, keep the base. So blood pH is going up. Everybody good? We're getting a few little frowns out
Well,
there.
is it going up or it's just regulating it, isn't it? Keeping
No,
it
it's
where
act-
it needs to be.
See, the problem is, we're making acid
Right.
all the time, or we're eating typically foods that make our blood more acidic.
Right,
And so we want to do two things, get rid of some of that excess acid
Right.
and neutralize the acid that's still in the
well,
blood.
that's what I'm saying, I mean, so it, depending on what's coming into your system, it's gonna be doing more or less of that to keep your pH where it needs to.
Yeah, then your normal pH is
That's-
7.35 to 7.5. So depending on what the pH is how active these cells are
That's
and
what
which
I
of these
meant,
cells are
that
active.
it's regulating it.
Yeah,
Okay.
it is being regulated. All right. We're doing really quite well. Now, type B. If you got type A, these type B, same ones, intercalated cells. The way to think of them is a type B cell is a flipped type A. Watch. We're going to take CO2 in, hook it up with water, same, everything's the same, form the carbonic acid. What's going to be the difference? Is the proteins that are on each of the luminal membranes is reversed. So what do we do with the bicarbonate by way of type B cells? We secrete that's the base, and we reabsorb.
So this is part of the regulation.
This is, yeah, so it depends on what's going on with your pH of your blood, which is, it's just amazing, we have both, right? Type A and type B cells. Type A is usually the one that's, so on your thing there for 5, what's the net effect for type A cell? What was the net effect? Reabsorb bicarbonate, secrete the acid, the hydrogen ions. Or, you know, it doesn't matter, say it either way. And then what's the net effect on 6 for type B? Reabsorb the acid, hydrogen ions, secrete the bicarbonate. You want to write that up there? I thought I saw a couple of, what did she say? So what's the net effect here? What do we do?
Secreted acid.
Secrete the acid, hydrogen ion, which is the acid,
Reabsorb the base.
reabsorb the bicarbonate, which is the base. So what's the net effect for type B? Secrete the bicarbonate, the base, and reabsorb the hydrogen, which is the acid. All right, so all that hopefully is making more sense than, you know, maybe when you were doing your lecture days. So here's what we got coming up. We've got a way back on page, we went through page, I think 12, for my notes. I think we're on 13 when we start next week on elimination of waste. So we got to still do the rest of this lecture, and we're also going to do fluid balance, so make sure you're ready to take your quiz and take notes on fluid balance
Well, so,
on Tuesday. next
so,
week.
okay, so you're saying the normal class quiz, we don't, is that what you're talking about, taking the quiz?
You gotta take your quiz, yeah, the normal fluid, it's a one on fluid balance, the lecture is scheduled for Tuesday.
Okay.
But we still, just don't forget to bring these notes, because we haven't
Right,
finished.
but I'm saying the quiz for this one is for today, it's due by Sunday.
Yeah,
We still, it's only up to this point?
oh, I see what you're asking, you're not asking me about the in-class quiz.
No.
No quiz.
I don't think I have anything on there relative to the rest of that lecture.
Okay.
Just maybe on elimination of waste on page 13, that's the only thing you should know about.
For DC2, could you tell me what you want us to know for number 10, urine?
Oh, yeah, if you had to describe urine, it would be plasma, right, the filter plasma, minus the plasma proteins, and then minus the nutrients, because they're 100% reabsorbed.
They're filtered plasma.
Filtered plasma, mine is the plasma
Mine is
protein,
plasma
so they're not filtered, to speak of. And then mine is the nutrient, because they're 100%. What I just told her?